Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. Sexual reproduction in flowering plants with monosporic embryo sac development. If you like this video, subscribe my channel and click on bell icon to get notifications. Like, follow and share my videos to your students and friends to get more information on biology related to NEET and other national state level competitive exams. In this video, I am going to explain the monosporic development of embryo sac because embryo sac is the main structure contains female gamete. The development of female gamete it takes place with the help of two steps in gynecium. In this, the first step is megasporogenesis and the second step is megagametogenesis. After completion of megasporogenesis, totally 4 haploid megaspores are produced from a diploid megaspore mother cell with the help of meiosis division every time. Out of these 4 haploid megaspores, either 1 or 2 or 4 megaspores, they acts as a functional megaspores to participate in the development of embryo sac or female gametophyte. Based on the number of megaspores participated in embryo sac development, the embryo sac developmental process is 3 types. In this, the first type is monosporic development. The second type is bisporic development. The third type is tetrasporic development. Let us discuss monosporic embryo sac development in this video. In monosporic embryo sac development, only one functional megaspore it is participated in the development of embryo sac or female gametophyte. This monosporic development it is again two types. The first type is Polygonum type and second type is Oenothera type. In Polygonum type of embryo sac development, only one functional megaspore is participated in embryo sac development, but the location of these functional megaspore in ovule, especially in nucellus tissue, is towards chalazal end. The chalazal megaspore it acts as a functional megaspore. The remaining 3 megaspores which are present towards microfilar end, these megaspores are non-functional in nature. These non-functional megaspores will degenerate. The Polygonum type of embryo sac development first time reported by Edward Strasburger in 1879 in one of the plant that is Polygonum divaricatum. So that this embryo sac development it is named as Polygonum type. The Polygonum type of embryo sac development it is the most common type of embryo sac development present in majority of angiosperms, nearly 70% of cases. In Polygonum type of embryo sac development, at beginning, the megaspore mother cell present in nucellus tissue, it is microfilar end and it is chalazal end. This megaspore mother cell it undergoes meiosis division to form haploid megaspores with the help of megasporogenesis process. During this, after completion of meiosis 1, two haploid cells are formed. It is called dyad. After completion of meiosis 2, totally 4 haploid megaspores are formed linearly, but in these megaspores, the three megaspores which are present towards microfile end, it become non-functional and it will degenerates. The megaspore present towards the chalazal side, it become functional. These chalazal microspore, now it participate in embryo sac development with the help of free nuclear divisions initially. During this free nuclear divisions, one nucleus it becomes into two nucleus becomes into four, and four nucleus finally it becomes into eight nucleus. Now such type of divisions are called free nuclear divisions. After completion of free nuclear divisions, every nucleus it can synthesize a cell wall around it, then it becomes into cell. Now totally 8 nucleus are formed in functional megaspore. Then cell wall formation takes place with the help of cytokinesis process. After completion of cell wall formation, the three nucleus migrated towards microfilar and they can synthesize cell wall around it. The three nucleus which migrated towards chalazal end also can synthesize cell wall around it, but the two nucleus present at center they never synthesize any cell wall around it. Now it is organized into a haploid multicellular structure. Now it is called embryo sac or female gametophyte. In polygonum type of embryo sac, we can observe some important key features. The polygonum type of embryo sac is a 7-celled and 8-nucleated structure. In this embryo sac, the cells are arranged in 3 plus 2 plus 3 pattern. In this, the three cells which are present towards microfilar end, it becomes into egg apparatus. In this egg apparatus, at center one large cell is present. Now it is called egg cell. The two lateral cells are called synergids. In central cell, the embryo sac contain only two polar nucleus. Towards chalazal end, the three nucleus after synthesizing cell wall, it becomes into three antipodal cells. These are the main characters of polygonum type of embryo sac. Let us discuss the second type, that is Oenothera type. In this Oenothera type also, only one functional megaspore it participated in the development of embryo sac. In this, the functional megaspore, its location is towards microfilar end. This embryo sac development, it is observed in some of the members of Oenotheraceae family. This Oenothera type of embryo sac development, it was first reported by Hofmeister in 1849 in one of the plant, that is Oenothera lamarckiana. It is also called evening primrose. Because of this reason, this embryo sac development, it is named as Oenothera type. In Oenothera type of embryo sac development, the megaspore mother cell which present in nucellus tissue having microfilar end and chalazal end. This megaspore mother cell, it undergoes meiosis division for the formation of haploid megaspores with megasporogenesis. After completion of first meiosis division, it formed two cells. Now these are called diod. After completion of meiosis 2, the diod becomes into tetrad. Totally 4 haploid megaspores are formed linearly. In these megaspores, the 3 megaspores present towards chalazal end, they become non-functional, and the remaining 1 megaspore which present towards microfile end, it become functional. The non-functional megaspores will degenerate during embryo sac development. The functional megaspore, which is present towards microfilar end, they undergoes free nuclear divisions, and after completion of first division it form two nucleus. The two nucleus then it becomes into four nucleus only. This nucleus can synthesize cell wall around it, then it becomes into cells. Now it is the structure of embryo sac in Oenothera type. The main key features of Oenothera type is, in Oenothera type of embryo sac, it is four-celled and four-nucleated structure. In this, the embryo sac it shows 3 plus 1 and 0 arrangement. In the structure of Oenothera type of embryo sac, the egg operators which present towards microfilar end having 3 cells. In this, the central large cell is called egg cell and two lateral cells are called synergids. In this embryo sac only, it contains only one single polar nucleus in central cell. Towards the chalazal end, antipodals are absent. These are the main characters of Oenothera type of embryo sac. Let us discuss some main differences in between Polygonum type and Oenothera type of embryo sac. In Polygonum type of embryo sac, the megaspore which is present towards chalazal side, it is functional. But in Oenothera type, the megaspore which present towards microfilar end, it is functional. The Polygonum type of embryo sac development, the functional megaspore nucleus undergoes mitosis division 3 times. But in Oenothera type, the functional megaspore nucleus it undergoes divisions only 2 times. After completion of free nuclear division, totally 7-celled and 8 nucleus are formed in this embryo sac. But in Oenothera type, it contains 4 cells and 4 nucleus. In Polygonum type of embryo sac, the central cell contains only 2 polar nucleus. But in Oenothera type, the central cell contains only 1 polar nucleus. In Polygonum type, antipodals are present towards chalazal side. But in Oenothera type, the antipodals are completely absent. These are the main differences in between Polygonum type and Oenothera type of embryo sac. Both these are example for monosporic development. Thank you for watching. If you like this video, subscribe my channel, like and share my videos to your students and friends.